So I want you to imagine your immune system, right, as a highly trained SWAT team.
Oh, I like that analogy.
Right. Like they are armed to the teeth, perfectly capable of taking out rogue cancer cells and basically just ready to deploy at a moment's notice.
Right.
But there is a fatal flaw in their whole operation. They're completely blind.
And a way of blind.
They rely entirely on these like intelligence briefers to hand them a photograph and say, this is the target, go get them.
Exactly.
And if those briefers are silenced, or if they just stop handing out the photos, that SWAT team just stands around doing nothing. And the tumor is just free to grow.
Yeah, it is a chilling image, honestly, but it's the actual physical reality inside a growing tumor. I mean, the immune system has the firepower, but it just completely loses the intelligence.
Which is exactly why we are looking at this today. For this deep dive, we are unpacking this really groundbreaking paper from Science magazine.
Yeah, it's a huge paper.
It really is. And the goal here is to figure out exactly how tumors manage to silence these intelligence briefers and, more importantly, how a really specific drug can basically turn them back on.
Right. It's all about flipping this epigenetic switch to, make the immune system recognize cancers it was previously just totally blind to.
Exactly. So to understand the mechanics of this, I think we first need to establish the two main cellular players here.
Right. So first you have your SWAT team. These are the CD8 T cells, basically the cytotoxic assassins of the immune system.
Assassins, got it.
And second, you have the intelligence briefers. And these are a really specific, rare subset of immune cells. They're called CDC1s.
CDC1s.
Yeah, or conventional dendritic cells, if you want the full name.
Okay, so I know dendritic cells generally act as messengers, but why are these specific CDC1s so crucial when it comes to cancer?
Well, it's because they possess this really unique biological mechanism called cross-presentation.
Cross-presentation.
Yeah. So most cells in your body can only show the immune system proteins that were manufactured inside themselves.
Okay.
If a cell gets infected by a virus, it puts a little piece of that virus on its surface to say, hey, I'm infected, kill me.
So just a basic distress signal.
Right. But CDC1s do something much more complex. They actively scavenge their environment.
Oh, wow.
Yeah, they crawl through the tissue, find bits of dead cancer cells, consume them, break them down, and then they physically present those external tumor fragments, the antigens on their surface, to the CD8 T cells.
So they're basically out there scavenging the battlefield to find the enemy's uniform.
Exactly. And then they hold that uniform up for the SWAT team to see.
That is wild.
And without this specific cross-presentation, the CD8 T cells simply cannot initiate an attack against the tumor. They just stay completely dormant.
Okay, so if a patient has a growing cancer, there has to be a breakdown in this specific handoff, right? If the briefers are in the tumor, why aren't they doing their job?
Because tumors engage in chemical warfare. I mean, a tumor isn't just a lump of bad cells. It actively creates what oncologists call an immunosuppressive microenvironment.
Like a localized force field.
Kind of, yeah. It secretes this dense chemical soup. And one of the main ingredients in that soup is a molecule called TGFβ.
TGFβ.
Right. And when the CDC1s are exposed to high levels of TGFβ, they shift from being these active, highly mobile stimulators into this dormant, tolerogenic state.
So the tumor is essentially flooding the area with a chemical sedative. It's just putting the briefers to sleep.
That's exactly it. And the result is what we call a cold tumor.
Right. I've heard that term before.
Yeah, because the T cells aren't being briefed. They never actually infiltrate the tumor.
Oh, wow.
And this is actually the main reason why some patients don't respond to our best current immunotherapies, like the checkpoint blockades.
Right, PD-1 drugs and stuff.
Exactly. Those drugs are designed to take the brakes off the T cells. But if the T cells don't even know what they are looking for,
taking the brakes off doesn't accomplish anything. They're just sitting there revving their engines, but they don't know where to drive.
Exactly.
Okay, so let's do a quick checkpoint for you listing because this really sets up the entire problem we're diving into today.
Yeah, it's the core issue.
So the SWAT team is ready, but the briefers are heavily sedated by the tumor's chemical soup. If we want to fight these cold tumors, we have to wake up the briefers.
Right.
So if TGF-β is the sedative, how is the dendritic cell actually, drinking it? There must be a physical receptor or an internal mechanism that gets triggered to shut the whole cell down, right?
Right. And that physical mechanism is exactly what the researchers in this science paper hunted down.
Oh, nice.
Using this really advanced single-cell sequencing, they actually looked inside these sedated dendritic cells and found the internal brake.
And what is it?
It's an epigenetic enzyme called CARM1.
CARM1. Okay, I want to make sure I grasp the physical reality of this for a second. An epigenetic enzyme that doesn't change the actual genetic code, right? It changes how the code is read?
That is a great way to put it, yeah. CARM1, it stands for co-activator-associated arginine methyltransferase 1.
Okay, that is a mouthful.
It is, yeah, but the methyltransferase part is the key.
How so?
Well, think about how DNA works, right? You have roughly 6 feet of DNA stuffed into a microscopic cell nucleus.
Which is insane to think about.
Totally. So to fit, it has to be tightly wound around these little protein spools, which are called histones.
Okay, the schools.
Right. And CAR1 is an enzyme that attaches tiny chemical tags, methyl groups, to those spools.
Okay, so if the dendritic cell's DNA is this massive, detailed instruction manual for how to activate T cells and fight cancer, is CARM1 sort of like, I don't know, a rogue librarian who goes around gluing specific pages of the manual together so they just can't be read?
You're definitely on the right track there, but gluing pages implies it's like a messy accident.
No, it's not.
No, it's actually a highly organized physical mechanism. Think of it more like the librarian taking that instruction manual and wrapping it in heavy steel chains.
Well,
yeah, when CARM1 adds those methyl groups, it actually changes the electrical charge of the spools.
really?
Yeah, the DNA winds itself so tightly that the cellular machinery literally cannot physically access the genes required to sound the alarm.
Wow. So the instructions for how to be a briefer are still in the cell. They haven't been deleted. They're just locked away in a vault.
Exactly.
So I assume the researchers immediately tried to break the vault.
They absolutely did. They designed a brilliant experiment to see what happens if you remove CARM1 entirely from the equation.
How do you even do that?
Well, they used CRISPR gene editing technology to create a specialized strain of mice.
Okay, lab mice.
Right. And using a genetic tool called XCR1TA, which you can kind of think of it acting like a highly specific genetic sniper rifle.
Oh, the genetic sniper rifle. Okay.
Yeah. They deleted the CARM1 gene, but they only deleted it inside the CDC1 dendritic cells. Every single other cell in the mouse still had normal CARM1.
That is incredibly precise. So what happened to the dendritic cells when they, removed the chains from the book?
The transformation was profound. I mean, without CARM1, those dendritic cells just woke up.
They ship off the sedative.
Exactly. They became hyper-efficient at cross-presenting those tumor antigens and multiplying the killer CD8 T cells.
That's amazing. But I have to push back here for a second, though.
Sure.
If we are permanently altering the epigenetics of a major immune cell, doesn't that wreak havoc on the rest of the mouse? Like, if we permanently unchain the alarm system, does the mouse develop massive autoimmune diseases because its immune system is just stuck in overdrive?
It's a really vital question, and it's actually something they checked meticulously in the study. The answer is surprisingly no. Deleting this gene had zero detectable effect on the healthy, steady-state mice.
Wait, zero?
Zero. Their peripheral immune systems were totally normal. It didn't even affect the CDC1's closest cellular cousins. The effect of removing CARM1 was incredibly specific. It only unleashed the CDC1s that were actively infiltrating a tumor.
That is wild. It's like finding a master switch that only turns on the exact fire alarm you need without setting off all the other alarms in the whole building.
It really is. And this brings up the next layer of the research, which I find fascinating.
Lay it on me.
So we know knocking out CARM1 works, but the researchers wanted to understand the structural changes happening inside the cell. Like, how do we know which chains were removed from which books?
Right, because the cell has thousands of genes. How do we know what CARM1 was actually locking away in the first place?
Exactly.
Yeah.
To figure that out, they used a technique called multi-omic analysis.
Multi-omic analysis.
Yeah, and one half of this is something called ATAC-seq.
ATAC-seq.
You can basically think of ATAC-seq as a molecular mapping tool that finds all the open and closed regions of DNA.
Okay, how does it do that?
It uses an enzyme that physically cuts into open DNA. So if the DNA is wrapped in chains, if it's closed, the enzyme can't cut it.
Oh, that's clever.
Right. So by sequencing where the cuts actually happen, you get a physical map of the genome's accessibility.
So they mapped the DNA of a normal dendritic cell against one where CARM1 was deleted. And what did the map show?
It showed that when you remove CARM1, the chromatin, which is the packaged DNA, physically opens up at very specific critical locations.
Like it unwinds.
Yes. And these newly opened regions contain the binding sites for master transcription factors, primarily one called BTF3, along with another called NFKB.
Okay, let's avoid getting lost in the alphabet soup here. BTF3 and NFKB, why do these matter so much?
Because they are the ultimate master switches for the immune cell. BTF3 is basically required for CDC1 to even exist and maintain its identity.
Okay.
And NFKB is sort of the universal engine for cellular activation and survival in the immune system.
I see.
So by removing CARM1, you are physically unwinding the DNA right at the exact spots needed to turn the dendritic cell into a hyperactive immune stimulator.
Okay, let's pause and look at the bigger picture here, because I want to think about this from an evolutionary perspective for a second.
Sure, go for it.
If CARM1 exists solely to lock away the most important functions of a dendritic cell, why does the cell even have it in the first place? Is this just like a biological design flaw that cancer somehow learned to exploit?
It's definitely not a design flaw. It's actually an environmental sensor.
An environmental sensor?
Yeah, I mean, your immune system is incredibly dangerous. It needs a reliable way to gauge whether it should be active and fighting or resting, to prevent chronic runaway inflammation.
Oh, okay. So it needs a volume dial.
Exactly. And the paper demonstrates that CARM1 is the dial, and it's directly controlled by the chemical environment.
So going back to TGF-beta.
Right. Remember TGF-beta, the tumor sedative?
Yeah.
The researchers proved that TGF-beta directly increases the production of CARM1 to basically turn the volume down.
It tells the cell to go to sleep.
Exactly.
Yeah.
But on the flip side, say during a viral infection, your body produces stimulatory chemicals like interferons.
Right.
And the researchers found that these stimulatory chemicals actively inhibit CARM1 expression.
Ah, so it's a chemical tug of war.
Yes, exactly. CARM1 sits in the middle, integrating these opposing signals. So the tumor screams, go to sleep. By pumping out TGF-beta, CARM1 levels rise, the chains go on the DNA, and the T cells stay blind.
Wow. It's an elegant but kind of tragic hijacking of a normal cellular control mechanism.
It really is.
Okay, let's do another quick recap for you listening to just lock this all in. We've identified the internal brake, which is CAR1.
Right.
And we know it physically restricts access to the master switches of the dendritic cell by altering how tightly the DNA is packaged.
Yep, the chain's on the book.
Exactly. And we know it's being manipulated by the tumor's chemical environment. So, I mean, Petri dishes and genetic snipers are fascinating, but what happens when we take this knowledge and test it in a living, breathing tumor?
Well, this is where the biology really translates into real disease models. They tested this in mice using a colon cancer model and a notoriously aggressive melanoma model.
Melanoma is notoriously tough to treat, right? Because it's a classic cold tumor. It essentially just ignores the immune system.
It does. But when they implanted these really tough tumors into mice,
right,
where CARM1 was genetically knocked out in the CDC1s, the results were dramatic.
Really?
Yeah. They saw a massive influx of active CD8 T cells flooding into the tumors.
Yes. The SWAT team finally got their target photos.
They absolutely did. And these T cells were highly functional, too. They were pumping out aggressive inflammatory chemicals.
So what happened to the tumors?
The clinical result was a profound slowing of tumor growth. But perhaps the most important finding was that tumors that were previously completely resistant to standard PD-1 immunotherapy suddenly became highly sensitive to it.
Oh, because PD-1 inhibitors just take the brakes off the T cells, like we said earlier.
Right.
So once the dendritic cells were awake to give them the map, taking the brakes off finally worked.
Precisely. Now, there is this fascinating secondary detail in this paper that we have to talk about.
Okay, what is it?
Well, decades of prior oncology research actually established that CARM1 helps tumor cells thrive. Yeah, in a cancer cell, CARM1 acts as an activator for cancer-promoting genes.
Hold on. So CARM1 helps the tumor cell grow, but it stops the immune cell from fighting.
Yes. It plays dual, entirely opposing roles depending on which cell it's actually inside.
Okay, my mind immediately goes to the ultimate experiment here. What happens if you knock CARM1 out in both? Like you take it out of the tumor cell and you take it out of the dendritic cell.
Well, you'll be happy to hear the researchers ran that exact experiment.
No way! And
they implanted CARM1 knockout tumor cells into mice that also had CARM1 knockout CDC1s. And the effect was massive and highly synergistic. The survival benefit just went through the roof.
Man, think about what that means for a second. It's like fighting a war where, with a single action, you are simultaneously dismantling the enemy's armor by weakening the tumor cells while instantly upgrading your own army's radar and targeting systems by supercharging the dendritic cells.
It is a molecular double whammy. Hitting one specific epigenetic target cripples the tumor's defense and optimizes the immune system's offense at the exact same time.
That is just brilliant in theory, but I do have to play devil's advocate here.
So for it.
Genetic engineering in lab mice before they are born yields great data, sure. But how does this help the listener or, a patient sitting in an oncology clinic today? We can't exactly inject CRISPR into a patient's bloodstream and safely edit all the dendritic cells inside their body.
You're absolutely right. I mean, genetic knockouts are just the proof of concept. To actually treat a human patient, we need a delivery mechanism. We need pharmacology.
Right.
And that is exactly where the researchers introduced a small molecule drug called EZM2302.
EZM2302. It sounds like a serial number, but I assume it's a CARM1 inhibitor?
Yes, exactly. It's a chemical compound designed to slip inside a cell, find the CARM1 enzyme, and physically block it from adding those methyl tags to the DNA spools.
Okay, so how did they actually deploy it? Did they just inject it into the bloodstream like a traditional chemotherapy drug?
No, because they wanted to avoid systemic side effects. They did something much more targeted. They built a localized cancer vaccine. Yeah, and to do this, they used a biomaterial called a mesoporous silica rod scaffold.
Whoa, stop right there. Mesoporous silica rod scaffold. What does that actually look like if I'm holding it in my hand or if it's like inside a body?
Fair enough. That's a lot of jargon. Think of it as a microscopic 3D jungle gym made of harmless silica.
Okay, a jungle gym.
Right. It's highly porous. You inject this tiny jungle gym just under the skin. But before injecting it, the researchers coated this jungle gym with cancer antigens, the target photos, along with some chemical attractants to naturally draw immune cells in.
So it's basically like baiting a trap.
Exactly. You want the dendritic cells to crawl inside this jungle gym to find the antigens. But here is the critical innovation they made. They added the EZM2302 drug directly into the structure of this jungle gym.
Oh, so when the dendritic cells show up to the training camp,
they crawl into the porous structure, they encounter the cancer antigens, and simultaneously, the drug absorbs into them and physically blocks their CARM1.
That's incredibly smart.
Right. Because the CARM1 is blocked, they don't get suppressed by any of those tumor signals. The results were explosive. There was a massive localized recruitment of CDC1s.
They all flooded the jungle gym.
Yes. And the drug allowed them to mature perfectly, detach from the scaffold, and physically migrate through the lymphatic vessels straight to the draining lymph nodes.
Where all the T cells are just waiting around for instructions.
Exactly. And the end result was a huge spike in neoantigen-specific killer T cells circulating throughout the entire body. The vaccine provided the intel, and the drug ensured the briefers stayed awake and highly motivated to actually deliver it.
Okay, this all sounds incredibly promising, but I have to ask the ultimate question here. We've cured cancer in mice thousands of times in the lab, right?
Oh, I have.
But mice are not humans. Does blocking CAR1 actually work on human immune cells?
Well, the researchers knew this was the vital next step to prove any sort of clinical relevance. However, it is incredibly difficult to isolate enough pure CDC1s from human blood to actually run these kinds of tests.
So what did they do?
They did something really remarkable. They took human hematopoietic stem cells from cord blood and meticulously nurtured them in the lab over weeks, guiding them to develop into authentic human CDC1s.
Wait, so they basically grew human intelligence briefers from scratch in a dish?
Yes. And then they ran the tests on them. Whether they used CRISPR to knock out the gene or they treated the human cells with the EZM2302 drug, the results mirrored the mouse data perfectly.
Seriously?
Yeah. Inhibiting CARM1 in these human cells massively boosted their activation markers.
When you say activation markers, what does that mean physically? Is that like flags on the outside of the cell?
Precisely like flags, yes. To hold up the target photo to a T cell, the dendritic cell needs physical scaffolding and chemical grips on its surface to actually lock onto the T cell.
Oh, to physically connect with it.
Right. And when CAR1 was blocked, these human cells built vastly more of that scaffolding. And to prove it worked, they actually fed these human CDC1s a human melanoma antigen.
And what happened?
The CARM1 inhibited cells were vastly superior at presenting this antigen to human T cells. It caused the T cells to rapidly multiply and secrete those heavy-hitting inflammatory chemicals.
Wow. So they proved the entire mechanism all the way from a mouse's DNA up to human stem cells.
They did.
So what does this all mean for you listening? I think let's just summarize the core insight here. Because for a very long time, the billion-dollar immunotherapy industry has focused almost entirely on the T cells, right?
Mostly, yes.
We've been inventing new ways to take the brakes off the assassins. But this research proves that if we want to cure cold-resistant cancers, we have to look further upstream. We have to treat the intelligence briefers.
That really is the paradigm shift here. Because at the end of the day, the T cells simply cannot kill what they cannot see.
Right. By using a small molecule drug to flip an epigenetic switch, basically to take the heavy chains off the instruction manual, we can force the immune system to recognize tumors it was previously totally blind to.
Which is huge.
It is. This mechanism makes both future cancer vaccines and current immunotherapies just exponentially more effective.
It absolutely opens up an entirely new therapeutic axis.
Yeah.
But, it also leaves us with a pretty profound question about the fundamental nature of our immune system.
Ooh, okay. What's the question?
Well, if CARM1 acts as an epigenetic volume dial, constantly reading the chemical environment to suppress dendritic cells, it raises the possibility that we've really only scratched the surface.
How do you mean?
Think about it. How many other immune cells, like macrophages or natural killer cells, have their own hidden epigenetic dials just waiting for us to discover and drug them?
Man, that is a staggering thought, because if we can find those hidden dials, we aren't just sending in a SWAT team anymore.
No, we're not.
We're basically controlling the entire intelligence network.